Hi, everybody. Welcome. Thank you so much for joining us for Suds and Science. My name is Andrea Decker, and I have the pleasure of working for the Fleet Science Center. I'm happy to see some familiar faces and names. Hi, everybody. If you've been with us before, you know how this works. If you have not been with us before, Suds and Science is a monthly science talk that we normally host out in the community. Unfortunately, that can't happen right now. We normally go to restaurants and bars all over San Diego County, but we're super happy that we still have the opportunity to bring this program to you in the virtual world. It's better than nothing, I say. This way we can still get together, learn something new, and find out about the exciting work that is being done in San Diego. Suds and Science is not your traditional science lecture. It really is meant to be a talk, and that means that we start with a presentation by our speaker, which is Dr. Nicole Steinmetz today. And then that's about 25-30 minutes, and then we open it up for Q&A and discussion with the audience. So Nicole might have some questions for you. We hope that you have some questions for her, and then we can discuss what we've heard and find out a little bit more about the impact it might have for society and us. That's kind of Suds and Science in a nutshell. Like I said, normally we're in restaurants, so hopefully you're comfortable at home. You've got your snacks and drinks ready. I just finished up my dinner, so please feel comfortable. We're going to be recording the first part of this event, which is the presentation by Nicole. And once she's done, I turn off the camera, which I hope will make it a little bit more, sorry, I'm trying to make sure that I admit everybody in the waiting room, a little bit more at ease to turn on your camera. This is more meant as a conversation, so we hope that once we turn off the recording that you will join us with a picture so we can see each other and talk with each other. It's just a little bit more personal that way, but I understand the camera is off for while we record. I certainly get that. All right, and I think that's all I wanted to say and point out. And with that, I'm going to turn it over to Nicole. Oh, there's one more thing I wanted to say. So normally when we are in bars and restaurants, we do not allow for the speaker to have PowerPoint presentation. And that's because, first of all, we want to foster some conversations, but also not all the venues have the capability. So it makes it really hard, and we don't have a portable system that we can take. So we're normally not doing PowerPoint, but we are in the virtual world. So we're trying all sorts of things with this program. So Nicole actually has prepared some slides for us for her presentation. So that just as a little heads up. All right, Nicole, you're up.
All right, well, thank you. All right, well, thank you so much for having me here and for the opportunity to share the story about our research. And maybe to get started, let me ask you this. Did you know that some vaccines and cancer immunotherapies are made from plant viruses? So probably the audience would say no, or maybe even say, what? There's plant viruses, or plants have viruses too? Well, within the next 20 minutes or so, I'm hoping to convey to you that A, yes, there are plant viruses, and B, we are utilizing them as a nanotechnology. So we're taking viruses that infect plants and we are repurposing them for applications in human medicine. So applying them as cancer immunotherapies and vaccines. So I will ask you this question again at the very end of this talk. And I will ask, do you know that some vaccines and some cancer immunotherapies are made from plant viruses? And when I ask that, I want all of you to say yes. And it's the cowpea mosaic virus. So that will be the test in about 20 minutes. So this is a talk that would usually happen in a bar. I'm German, so I am prepared. Cheers, everybody. And I figured I start out by showing you this picture of this virus. This is a transmission electron micrograph of the Acidianus bottle-shaped virus. And I want you all to very, very carefully focus on this picture, because then you can see what these things truly are. So these are teeny tiny beer bottles. But I'm just joking. These are just viruses that occur and have a really fantastic shape. And this is the perfect seminar to show these pictures. Let me start out by just saying a few things about myself. So you already learned I'm German, and you probably figured this was not the typical California accent. So I was born and raised in Germany. During my undergraduate degree, I already started working with plant viruses. So today my research lab utilizes plant viruses as a nanotechnology, and you're going to learn about this in a few minutes. But even during my undergraduate research, I've already worked with plant viruses. At that time, it was the potato virus X. And I've been fascinated from my undergraduate days with the opportunity that biology gives us. Biology gives us all sorts of cool materials, including viruses, right? Some viruses, this might be like dangerous to say when we go through a global pandemic, right? Viruses as cool materials. We all think about viruses and we don't necessarily think of them as cool materials. But some viruses are not dangerous to humans. They're non-infectious towards humans, but they have properties that are really intriguing to be utilized and repurposed for human medicine. And that's what I'm going to show you in a little bit. So in my early days, I already worked with plant viruses in a biotechnology setting. I did my PhD in England, where I continued working with plant viruses more in a materials setting. I asked questions like, can we organize them in 3D space? Can we use a plant virus to make a diagnostic sensor? So using just the features of the size of the virus to build new materials. I came to the US in 2007, where I joined as a postdoc at the Scripps Research Institute. Postdoc is a training that PhDs often do after their PhD to learn a new skill, a new technique. And this was for the first time when I thought about not just plant viruses as materials, but plant viruses could be utilized for human health applications. And that has become the theme of my own research lab, which I started in 2010. And then in 2018, we relocated the lab to the Department of Nanoengineering at UCSD. Being a scientist and being an engineer is tough at times. I've managed to build a very large program. We are very well funded, and I have a fantastic team of students and postdocs. But going through these different steps, I've also learned you fall down a lot, right? You start a new experiment and it just doesn't work. You write your first grant proposal to fund your research, and the reviewers will slap that back in your face and say, no, that's not a great idea, or that doesn't work. You submit your first research paper and your peers will review this very critically and send it back to you. But none of this has ever stopped me. And this comes actually from my earlier career as a high-level athlete. So when I was 13, I was drafted for the German national team in roller figure skating. And I competed for the German national team for about 10 years. And trust me, being a figure skater, you fall down a lot. And during this time, though, I've learned you always stand up and you try again until you succeed. And I felt when I entered graduate school, and also when I became a professor, this falling down and standing up has actually given me an advantage, that I had already learned to stand up and just to keep trying. And these setbacks that you feel as a scientist, I never saw that as a setback. I always just saw that as the next challenge. I have to convince them better to give me the research funding to go on. But maybe it was this, enough about me, and now going into the research. So being an athlete and then being a trainee and studying in different countries and in an interdisciplinary setting, I became to start my own research lab was this idea that we're going to use plant viruses as nanotechnologies, and we will repurpose them for applications in nanomedicine. So now let me step back. What is nanomedicine? What exactly is nanotechnology? And what do plant viruses have to do with all that? Well, first, nanomedicine, maybe a term people have heard. The question is is this fiction? Is it science fiction that we build these small machines that can go through the body and then repair or deliver drugs? The answer is no, it's not science fiction. It is a clinical reality. And maybe I didn't define what nano actually means. So nano is a prefix, comes from the Greek language, and mathematically it essentially means 10 to the minus 9. That might not mean much. Nano means dwarf, so it's very, very small. If you want to pull out one of your hairs and take a look at it, you can still see the hair with the naked human eye, or like me, maybe through your glasses. But it's a small object that you can see. A nanometer object is 2,000 times smaller than the width of a human hair. So very, very small, small enough that it could be injected into the bloodstream, into the body, small enough that it can navigate the body, small enough that it can interface with the human body, that it can interact with cells, that it can enter cells. And this is no longer science fiction. It is a clinical reality. And nanomedicines have been approved back in 2005. So for many years already, we're using nanoscale particles as medicines in various settings. Many examples are for cancer treatment, where we use lipid nanoparticles, essentially fat nanoparticles that encapsulate these toxic drugs that are given to kill tumor cells. And this nanoscale delivery allows to reduce some of the adverse side effects that patients feel when they have these treatments, but it also allows to interface with the body better. And I will get into this in the next few minutes. And then, of course, many of us maybe already have been vaccinated. Moderna and Pfizer are lipid-based nanotechnologies delivering COVID-19 vaccines. AstraZeneca and also Johnson & Johnson approved now are viral vectors that are delivering vaccines. And then our technology is not yet clinically approved, yet we're on the way to get into clinical trials, would be plant virus-based nanotechnologies. I want to show you, I will go out of this presentation for just a minute, because I wanted to show you a video that just kind of highlights why nano is so important in medicine. And you should be able to see this video now. It comes with some fancy music too. Can you hear the music as well?
Yeah.
So I will just let this run maybe for 30 seconds, and then I will start talking again. Gives me some time to have some beer. So everything you're looking at are nanoscale objects. This, for example, is a lipid raft. It's an object that floats on our cell surface, and it harbors these three objects kind of sticking out. These are receptors. Receptors are proteins, nanoscale proteins that are kind of sitting on the surface of our cells. They are nanoscale objects, 2,000 times smaller than the width of a human hair. And these objects, as you can see here, they can grab onto other small objects, like a cytokine shown here. A cytokine is a molecule that cells use to communicate with each other. Like one cell will send out a cytokine to tell another cell to do something. So the nanoscale is so important because that's how our cells communicate with each other. That's how our cells function. And that's how nanoscale materials can nicely interface with the biological system. That's why nanomedicine or nanoscale materials and medicine have made such an impact. And I will go back into my presentation now. The point that I wanted to make with this video, and I'll be happy to share the full length, the link with everybody. This is a video that is published on YouTube, so everybody should be able to find this. Okay, so you should be able to see the presentation again. Okay, perfect. So this is the video shows the inner life of a cell. And the point that I wanted to make here is our cells operate at the nanometer size scale. So if we can devise nanoscale particles, that means we can directly interface with biology. And not just to deliver drugs and therapies in a better way, but we can use nanoscale objects as the therapy itself by interfacing with the human body to restore normal function. And that will hopefully make sense in just a minute. So many different nanomaterials are being developed. Many have been already, or some have been already approved for clinical use, and many more in the clinical pipeline. So my lab has turned towards plant viruses, and I think there's many good reasons why we've chosen a plant virus over a synthetic material or a mammalian viral vector. So compared to the mammalian viruses, the plant virus offers safety. Plant viruses are non-infectious towards humans, and they're non-infectious towards mammals. So they are safer than using a mammalian system. But also when it comes to manufacture, they are easier to produce than a synthetic nanomaterial. And again, that will depend on the material you're using, but we literally grow these things on trees, or we literally grow these things in plants. And you can see here's a video I took of one of my students who's taking a solution of plant virus, just in a buffered solution, and gently rubbing it onto leaves. What happens then is the leaf becomes infected, the whole plant becomes infected. And as the plant grows, it's producing these virus particles for us. So again, we're using biology to make materials for us. This is very high yielding, it's very green. We don't use any toxic solvents or energy consuming heat processes to make these materials. And there's many more sort of reasons why we've using a plant virus over other materials, and I'd be happy to go into this more during the Q&A. So now I want to show you an application where we've used a plant virus to successfully treat cancer in dog patients. Like any research, we've done the in vitro testing, we've done all the cell testing, we've done preclinical studies using mouse models, and we have already advanced this into treating pets, dogs that have been diagnosed with melanoma and sarcoma. And we have done so very successfully. But before I show you, well, I'm actually not going to show you any data because I was told not to do that. But before I go into the story any further, I want to teach you just a very quick immunology lesson, Immunology 101. So everybody stay seated. So immunology is not an easy topic to teach to anybody, and I don't know what your biology background is. So I thought I'd ease you into this by showing you this very relaxing picture of this person walking on the beach. But there's a twist. This person is now going to step into a small wooden piece on the beach, and that wooden piece was infected with bacteria. Oh no. But no problem, because the person is a healthy person with a fully functioning immune system. So as the skin is poked and bacteria entering under the skin, immune cells that are sitting within the tissues will quickly realize there's a bacterium, and they will start eating up the bacterium, which makes them secrete cytokines, these small nanoscale proteins that are being sent out. What then happens is the blood flow suddenly is increased. The blood, that's when your toe becomes red because there's more blood flow. Other cytokines are being released that make the vessels leaky. So now you have the storm a local storm of cytokines, chemical molecules and proteins that allow cells to communicate, and any immune cell flowing by will get alerted. Oh, there's a problem there. I must exit. The vessels are already leaky. I will exit. I go into this tissue and I help with the fight. So that's why your toe becomes swollen. That's why it's red. You have more action there. The immune system will take care of this. Once the initial battle is over, these immune cells will traffic to the lymph nodes. Right? We all had a cold at some point. You can feel like your lymph nodes maybe slightly swollen. That's where a lot of action happens. That's where two specific kinds of immune cells meet. The innate cell, the initial fighter, meets more specialized immune cells, and it will tell them, look, there's bacteria in the body. This is what they look like. Go through the entire body and make sure there's no bacteria left. And so these specialized fighters now go and look for bacteria. So that's a very sort of simple example. We all had at some point, right, a splinter in our fingers and our toes, and there was no medical problem because our immune system already took care of things. The same mechanism works against tumors. And probably our immune system daily fights tumor cells, a cell that starts being abnormal. And the immune system will quickly realize this and quickly kill the cell, alarm other cells to kind of look, is there anything else like it? And the immune system can and will take care of tumors. However, very aggressive tumors find ways to overcome this. And before I go more into the problems, I restate what I just stated was the example of the bacterium entering your skin. I'll show you this cancer immunity cycle, which is the body's defense against a tumor. So in step one, you have a tumor somewhere. A tumor cell undergoes apoptosis. It undergoes cell death. And that can happen for all sorts of reasons. It ran out of space. It ran out of nutrients. It ran out of oxygen. So the cell simply dies. When this cell dies, if any cell in the body dies, there are these immune cells that are sitting around in our tissues and they're waiting for something to happen. So now there's a dead cell. They get excited. They go over there, swallow up the cell, and then they realize, you know what, something isn't quite right here. It looks like a normal human cell. It doesn't look like a bacterium, yet something is different. Maybe a protein looks different, or there is a protein that shouldn't be there. Something is wrong. So this immune cell will get fired up, traffic to the lymph nodes. Again, your lymph nodes, you have many lymph nodes in your body, but these are the ones that most of us are aware of. And will traffic to the lymph nodes and talk to these other specialized immune cells. In this case, the cytotoxic T cell, the little blue cell. And it will tell the cell, look, this is a tumor, go around the body and kill tumor cells. And the cells will go patrol the body and cancer never happens. So what happens is the tumor will fight against the fight, right? The tumor will tell immune cells that are coming in, kill me not. And they do this again through these nanoscale objects on the cell surface. They will literally talk to the immune cell that comes in and say, no, there's nothing to see here, kill me not. And the immune cell will let go. Or it will secrete out these chemokines and cytokines, the small proteins that will then prevent from these immune cells coming into the tumor. And so we call this, it's an immunologically cold tumor. It's cold because there are no immune cells, and the immune cells that are there are deactivated. So what we thought to do, and this is now where the plant virus comes in, can we relaunch the cancer immunity cycle? Can we tickle the immune system within the tumor to restore the cancer immunity cycle? So we are not coming in with a therapy to kill the tumor. We are really just coming in to see, can we relaunch the cycle? And the idea, I think, is beautiful, and it's also very simple. So we're taking a plant virus, and a plant virus is non-infectious towards human cells or mouse cells, any mammalian cell. But the plant virus is a foreign object. So it's recognized by the immune system. And it actually has all the bells and whistles to be recognized by the immune system because it looks just like a virus. It looks very similar to a mammalian virus, but it doesn't have the functions of being one. So what we've done is we take these particles and we inject them directly into the tumor. And what happens then is an immune cell that is there will think, oh my God, there's a viral infection. I need to call on all my friends. And they're not going after the tumor initially, they're looking for a viral infection, yet there's none. So in turn, they start attacking the tumor cells. And as they do this, they then also realize something isn't right. So they traffic to the lymph nodes. They activate these other immune cells. So if we have a patient, and whenever I say patient, I'm either referring to a mouse or a dog. When we have a patient with two tumors, say a dermal melanoma and lung metastasis, we only need to treat the dermal melanoma, the one in the skin, the injectable tumor. But because we are launching this cancer immunity cycle, and we are launching these immune cells that can go everywhere within the body, both tumors will disappear, the tumor on the skin and the tumor in the lung. And I'm not going to show you any data. I'm just going to show you these two cute dogs, Paige and Boo, which were both cancer patients. They were both treated. We have treated over 25 canine cancer patients to date. In all cases, the treatment was very successful. So many of these patients came in with large dermal melanomas, with oral melanomas, some had sarcoma, inflammatory breast cancer, and all patients have responded to the treatment very well. Paige survived for another 3 years after treatment. Many of these dogs are old, so she died due to non-related reasons. And many of these patients have done really well, better than they would have done on a standard of care. So based on these very promising results, we want to move this forward into clinical trials and to human clinical trials. And our target is to achieve that by 2022. So I want to use maybe the last 5 minutes to kind of switch gears just for a few minutes and tell you about how we went from a cancer immunotherapy to thinking about COVID-19 vaccines. So last March hit, it was about like a year ago, and everybody's sent home, right? Because of the SARS-CoV-2 virus, COVID-19. And every researcher around the world went home and started thinking about what can we do? And you see in the literature a lot of technology being pivoted towards COVID-19. Researchers being focused on, say, cancer treatment or on diagnostic tools, not working in infectious disease, but now applying their skills and tool sets towards solving a global problem. So what we started thinking about were vaccines. We've been working on cancer immunotherapies, so it was natural for us to start thinking about COVID-19 vaccines. And this is sort of where we go back to this initial picture that I showed you. And our idea towards the COVID-19 vaccine is sort of depicted here. So the idea is we use the plant virus as our core. The plant virus can activate the immune system. And then we modify its exterior to make it look like COVID-19 or make it look like SARS-CoV-2. So the virus itself is non-infectious, it's a plant virus, but we're taking fragments from this S protein, the red protein, the spike protein. We're taking fragments from there and we're putting it on the surface of the plant virus. So now when we're injecting it into, we're currently testing this in mice, when we're injecting this into mice, we're inducing immunity against SARS-CoV-2. And we have tested this. We took sera from these mice. So we collected blood samples from these immunized vaccinated mice. And in collaboration with the NIH and their BSL-3 labs, we have now tested that these blood samples from the mice neutralize the SARS-CoV-2 virus and protect human cells from getting infected. But we want to bring this even a step further. So, of course, many vaccines are already being rolled out. We are still in preclinical testing. Are we going to make an impact? I don't know for COVID-19. We're probably too late. But what our goal is to develop this platform technology so we are ready for whatever the next thing is that's going to hit us. And we have some unique advantages. The plant virus is stable in various temperatures and various environmental conditions. We don't need ultra-low freezers to store these samples. Another very attractive feature is that the plant virus is so stable, we can integrate it into materials. Shown here is a microneedle patch. And I actually have one here at home. So this is a small patch, a Band-Aid that has very small needles. And the idea is this can be shipped at room temperature. So this is an empty one. I'm not doing experiments at home. But then the idea is we can ship this to people's home, and just like a Band-Aid, you apply it on your arm and you can vaccinate yourself at home. This is currently not feasible with the lipid-based nanoparticle vaccines. There's stability issues, but the plant virus is very stable. So we have the advantage of generating technologies like this. So even though vaccines are being rolled out already, we are very eager to push this also towards clinical trials. So if we can demonstrate safety and efficacy with this platform, whether it's needed for COVID-19, I don't know, but it certainly could be very useful in the future for the next infectious disease. So I'll wrap this up now. I hope what I could convey to you is that, well, plant viruses are materials. They're very small materials, and we can repurpose them for applications in human health. We in my lab, we work on cancer, cardiovascular disease, infectious disease. We also have a program on using plant viruses for plant health. So maybe counterintuitive to use a plant pathogen to treat a plant disease, but we have an angle there. And yeah, I can't end this without thanking all my lab members. They're the ones doing all the hard work. I'm the one just doing all the talking. But it's a really fantastic team of people from all over the world at various levels: scientists, postdocs, graduate students, undergraduates, many collaborators within UCSD, and quite diverse. Right? This is a team effort. We're working together with material scientists, polymer scientists, with the formulation of these vaccine patches. We're working with clinicians, with immunologists. In particular, I would like to mention as Jack Hoops and Steve Firing, who was the immunologist and the vet who we're working with on the cancer immunotherapy treatment and our funding sources. And thank you very much for your attention. I'll be happy to answer any questions.
Nicole, thank you so much for an amazing presentation. I loved it. That was great. If you wouldn't mind unsharing your screen.
Yes.
Thank you. And then with that, I'm going to stop the recording. And for everybody or anybody watching this on YouTube later, thanks for watching it. And I hope you consider joining us in person, because now you're missing the conversation, the Q&A with the speaker. So you have to join us live to get the benefit of that. But thanks for watching the video, and I hope you enjoyed.